reputation of starting on time, and we're also going to aim to end on time and stick to the schedule throughout the 2 weeks. So welcome. We're thrilled that you're all here. You should have gotten an email this morning that Rosie and Hui sent from the Summer Institute email address about the computational problem sets. And we're also going to be posting problem set 2 today, so keep a lookout for that. So what I'm going to do in this talk is three things actually. First, I'm going to make some comments about the 2 weeks as a whole, and what this is all about and what you can expect. Then there's going to be a bit of substantive content about how to think about what genetic effects are. So that's going to be mostly review from problem set 1, but I think it'll help set the stage for the other talks that are coming later today. And then the third thing will be an outline of the syllabus overall for the next 2 weeks and kind of put things in perspective of how they relate to each other. And by the way, all these slides are in the Dropbox folder. So despite our technical difficulties, if you want to see the slides that I'm speaking from, you can open the PDF. They're not very interesting, actually. They're just bullet points of things I'm going to say anyway. So it's not that. This is the perfect lecture for us to lose the visual. All right. So we're very excited. This is our third Summer Institute in Social Science Genomics. We think it's been getting better over time as we've been getting feedback from students and adjusting accordingly. But nothing is ever perfect, and we want to continue to get feedback and continue to improve. So first of all, let us know throughout the 2 weeks. Let us know if you have thoughts or suggestions about ways we can make things better. And if you're too embarrassed to tell me or David or Patrick, feel free to tell Rosie or Hui, and they'll anonymously relay the message to us. We said some of this last night, but there's a number of people and organizations to whom we're extremely grateful who are making this event possible. Of course, the Russell Sage Foundation that funds the Summer Institute and created it. My wife, Samantha, who handled logistics, and Maribel Estrada, who you're not going to meet, but she dealt with administrative things on the back end at USC. The National Adolescent to Adult Health Study, AdHealth, who were very kind to make the data available that you are going to be using to do the computational problems over these 2 weeks. And by the way, it's a very unusual arrangement for you to have access to full individual level genetic data for a training course like this. So it's very unusual. Typically in these kinds of training programs, what happens is you get kind of fake data to use. We basically were able to get permission through the IRB, and actually the National Bureau of Economic Research is another group that we're grateful for. They provide the servers and also the IRB oversight that allowed us to get the AdHealth access. And finally, we're very grateful to the instructors who are volunteering their time to make this course possible. This Summer Institute is about social science genomics. What is social science genomics? It's the use of genomic data in social science research. And we believe that there's a lot of value to incorporating genetic data, genomic data in the social sciences. That there's a lot of payoffs. It's going to improve the way we can do social science. And a number of the lectures today are going to be about that, and you're going to see lots of examples throughout the course. At the same time, I think it's very important that we recognize from the outset, and we'll do this repeatedly throughout the 2 weeks, that there are particular challenges when we talk about combining genetics and social science. There are always challenges with any kind of science in terms of interpretation and communication and the appropriate use of the research, but those challenges are especially acute in this particular area because, of course, there's a terrible history of mixing genetics and social policy and eugenics, and unfortunately, in today's social environment, there's a rise of racism and a kind of new eugenics, and so we need to be hyper aware of that and make sure that that we are very careful in the way we interpret and communicate research in this area. So Michelle Myers is going to give 2 lectures that address these issues specifically, but much of the rest of the content of the program is also going to be about conducting good, responsible research. You can come up and try to set this up while I'm speaking. Thank you. Yeah. Okay, so what's special about genetic data?
Sorry.
So genetic data is different than a lot of other data that we have access to in the social sciences for a number of reasons. One is it's fundamentally what makes us different from each other. So to the extent we're interested in individual differences across people, you can't get more basic than DNA. It's a predetermined variable in the sense that no choices that you make throughout your life can affect your DNA. It's fixed at the moment of conception, and so that means that that we have a better chance of being able to answer certain kinds of causal questions with genetic data than we could with other kinds of data. We still are going to need to be very careful about inferring causation for a number of reasons we're going to talk about, but the fact that it's predetermined is helpful. So another very important feature of genetic data that's going to come up repeatedly is that it's randomly assigned, conditional on your parents. So given 2 parents, the DNA of the different children that they have are random draws from the DNA of their parents. That's another very useful attribute of the data in terms of learning about causation. Another interesting feature of genetic data is that it is finite. It's large, it's big data, but it's finite and fully measurable. In principle, we could have an individual's full sequence of data, and there's no concern that we're missing anything. That's almost never true in the social sciences. We're always worried about unobserved environmental variables. Entire genome. That would be all the genetic data that there is on that person. There's no omitted genetic data. We know that would be everything.
But in principle, we could observe every minute of a person's life cycle.
But how would we code it?
Okay.
Yeah, so we just don't know. I don't think we're ever going to be at the point where we can fully.
No, it's the coding.
Yeah. Another important feature of genetic data is that the biological functions of the genes are themselves, in principle, fully knowable. So that means that at least in terms of, we don't. I should be clear. We don't know about a lot of the biology of a lot of genes, but we're learning more and more at a very fast rate. And the fact that you could eventually know.
Sorry, I meant to borrow that at some point.
Sure. Yeah, go ahead. Sure, thank you. So the fact that we could know all of the biological functions means that we again have a chance of understanding causation, the mechanisms of causation, better than we often do. Now again, we have to be careful because often for the kinds of behavioral traits we're interested in as social scientists, the distance between the biology and the behavior is very far. So understanding the biology may not be that useful in terms of understanding mechanisms for the ultimate behavior. But it definitely is an advantage. Another important feature of genetic data is that it's a leading example of big data. So even if you weren't interested in genetics, and I think you all are, because we admitted you partly on that basis, the methods and approaches that you learn and need for genetic data are transferable to other uses of big data. OK. So the point is that genetic data has a number of distinctive, well-understood features that are useful for the purposes we're going to use it for. OK, so why? So I will argue that you are coming to the Summer Institute at the ideal time to be learning about applying genetics in the social sciences. Why do I say that? Well, there have been very rapid advances in terms of our ability to measure genetic variation. And that's largely because of the fall in costs. So if you could see the slide, there's a time series where it shows on the x-axis years and on the y-axis cost in terms of dollars, which is on a log scale. And there's a line that shows Moore's law, which is the famous rate of decline of the cost of processing power, which is very fast. And genetic data measuring genetic data is getting cheaper faster than Moore's law. It's now, we couldn't measure genetic data very well at all when the Human Genome Project was completed. It was very expensive in the year 2000. Today, the kind of data that we're going to be using, genome-wide data, which isn't quite sequencing data. It's not, we'll talk more about exactly what it is, but it captures much of the variation across people, and it can be measured at a cost of $30 a person, and from a saliva sample. So because of that fall in costs, genomic data is widely available in both the data sets that we use to learn about links between genes and traits, and also in the social science data sets where we'd want to use genetic data. Because of the growth in the size of the data sets for genetic discovery, we're now able to predict a number of phenotypes of interest to social scientists in some cases, at reasonably high levels, and in many cases at growing levels. So in the next couple of years, we're going to be able to construct polygenic scores for many phenotypes at levels of predictive power that will be useful in the social science data sets that have genetic data. So the time is really ripe now for applications. There aren't that many yet, but there is the possibility of many applications of genetic data in the social sciences, and not that many social scientists know how to do it. So now is the ideal time because you're going to learn the tools that will allow you to make these important early contributions in a field which is growing quickly. So I wanted to say a bit about who we are, those of us who are teaching at the Summer Institute. We represent a range of different disciplines. They include economics, sociology, psychology, genetics, and bioethics. The instructors are among the leaders in this field of social science, genomics, and several of us have been involved with the Russell Sage Foundation. In a number of capacities, as members of working groups of the Russell Sage Foundation, as grantees. David Cesarini and I are both alumni of the Summer Institute in Behavioral Economics. That is the longest running Summer Institute at the Russell Sage Foundation that Jim mentioned last night. And as side note, as Jim mentioned last night, the Russell Sage Foundation offers a number of grants and has a visiting scholars program, and that's something you may want to think about as you go forward in your career. Some of us who are instructors are core members of the Social Science Genetic Association Consortium, which is a large cooperative enterprise of researchers from different disciplines that do genome-wide association studies of behavioral phenotypes. So we create research tools like polygenic scores for the community of researchers. We aim to promote good research practices, like doing power calculations before you run a study, pre-registering your hypotheses, completely reporting your results, publicly posting results. So you'll hear a lot about those kinds of research practices throughout these 2 weeks. And we're committed to the accurate communication and interpretation of research findings. All right. So the three of us who are organizers, it's myself, David, and Patrick. All three of us have PhDs in economics, but all three of us also have a lot of involvement and engagement with other collaborators and colleagues from other disciplines. Okay, so I want to say a bit about who all of you are. You heard last night about you specifically, but to give you a kind of overall picture, you are 29 participants in total. Actually, yeah, so 29 participants out of a total of 56 applicants. It was a very strong pool of applicants, so it was extremely difficult to select 29. The reason why we chose you was we were looking for applicants who we thought were likely to become future leaders in the field. So, that's our hope for you.
No pressure.
Yeah, no pressure. There are, in terms of disciplinary breakdown, the largest single group is economics. There's 10 economists, but economists are the minority. There are 8 sociologists. That includes demography, 2 from statistics, 3 from psychology, 2 from biology, 2 from epidemiology or public health, 1 from psychiatry, and 1 from geography. So we think this interdisciplinarity of the program is a great strength, and I think you'll learn a lot from talking to each other and hearing different perspectives. There are a number of goals that we have for the Summer Institute. One, maybe the most important, is to help you develop your own research ideas. So we want you to be thinking about that throughout these 2 weeks, as you do the readings, as you're in the lectures, and then at the last day of the Summer Institute, there'll be time for you to make presentations of ideas that you've had, then get feedback from the group. So think now, starting now, toward the end, that last day, about what you might want to talk about. And of course, we encourage you to collaborate with each other. Another goal is for you to learn how to implement methods that use genomic data and interpret the results of those methods. So one of the things in particular that we think is relatively unique about this training program, as opposed to some other training programs that also teach some of the software for analyzing genetic data, is that we're going to talk a lot about what's underneath, like what the statistics underlying the methods are, where it comes from, and how to interpret the results. So it's not just going to be teaching you how to type out lines of code, but to understand what's being produced by that code. Another goal is to help you become a better consumer, more discerning consumer of research in this field, and in particular to understand more deeply some of the challenges to doing the research and interpreting the research. We're also hoping you're going to learn about some useful data and some software. So for example, the ad health data is data you may want to use in your own research. And so you'll get actual practice with using it, and you can then apply for it yourself to continue using it after the Summer Institute if it's going to be useful. And finally, learning about good research practices, which we think are important well beyond the area of social science genomics. Let me say a bit about our philosophy, our teaching philosophy for the Summer Institute. The field of modern genomics is an empirically driven field. There's just new discoveries all the time. It's very fast moving, and it's empirical. And that's why we worked so hard to get you access to real data and to the hardware and software that you'll need to be able to analyze that data. At the same time, we think that the core theoretical ideas are absolutely essential. So they're essential to identifying what are good research questions and what are good methods for addressing those questions, and they're crucial for interpreting the findings that come out of the research. And so that's why our emphasis throughout is going to be on the key theoretical ideas, which we're going to try to convey mostly through the problem sets, but also some in lectures through simple models that get across those key ideas. So that's why the problem sets are really central to the program. And we understand that that you come in with a variety of different mathematical backgrounds. So we don't necessarily expect that everyone is going to be able to easily answer all the problems. But we want you to engage with those problems and struggle with them if you don't know how to answer them. Work with other students who may have more background in a particular kind of area in order to solve them. And then go over the solutions. We're going to hand out the solutions right away when the problem sets are due. So we don't have the resources to grade the problem sets ourselves, but we want you to grade your problem sets and compare them with the solutions and figure out the right answers. And we think that there's really a big payoff to doing that. We've tried, to the extent we could, to have a uniform notation across the lectures and the problem sets, across different instructors. We can't do that perfectly, given the number of people involved, but hopefully it will help understanding that we've tried. Okay, so the Summer Institute is going to be intense. We think of it as an immersive learning experience. All of us instructors have put our own research and other commitments on hold for these 2 weeks to be dedicated to this experience for you. You probably found that problem set 1 took a long time. The other problem sets will also take a long time. There will be many hours of lecture a day. There is a lot of reading that you could do. And we also want you to be spending time brainstorming research ideas. So it's enough to more than fill up every day of the 2 weeks. And we hope you do that and get a lot out of it. In particular, I encourage you to take advantage of the instructors being around. We have some time specifically set aside for office hours, and we'll tell you where we're going to be. But also, we're around for meals, and we're living here with you. So pull us aside and talk to us if you have questions or ideas, or just want to talk. Our goal is to get to know. One reason why we try to have a relatively small group is so we can get to know all of you well. We encourage you to work on the problem sets in groups and meet each other and think of each other as potential collaborators, not just during these 2 weeks, but potentially also for after that. So a couple logistical notes. Everyone's expected to participate in all the activities over these 2 weeks. Every meal is provided by the conference center, except over the weekend. So starting Friday at dinner, there won't be meals. You'll be on your own for Friday dinner and through Sunday lunch. And then Sunday dinner, we'll be back here again. Lunch today will be where we had dinner and breakfast. That may change. It may end up being in this room on subsequent days, but we're still working that out with the hotel. So you know Samantha is the contact for any logistical questions, and you can always reach us at the RSF email address. And the slides give Samantha's cell phone and my cell phone if there's any emergencies. Okay. So let me skip the slide about the problem sets. I've already said a number of things about them, but I should also emphasize that Hui and Rosie, who are the TAs, are totally on top of all of the problem sets, both the more the analytical problems, but also especially the computational problems that they've created a number of the problems themselves and written the solutions, and so you should feel free to consult them heavily for any help that you need. Regarding the computing, you should have access, or you will have access very soon, to the cleaned data from AdHealth. So that we've cleaned for you, so it's ready to answer the problems. But you'll also have access to the raw data, which is what you would download if you got access yourself to the AdHealth data. You would get the raw data, and we provided you with the code that we use to clean the data, so you can see, the kinds of things that you need to do yourself to get to the clean data. We also provide for you the AdHealth codebook, so you can look up what all the variables are, including variables you don't need for doing the problems that we're asking you to do. And you should feel free, and we encourage you to explore the data. And if you have research ideas that might use the AdHealth data, you can start exploring them yourself here. And you have access to our server at the NBER that has the data and also software that you can use to analyze the data. And it's a powerful computer. So we can run the software that you would need. Hui and Rosie are going to give you your password for the server. They're going to give it to you only in person and on paper. So do not store it electronically. This is part of the data security for AdHealth that you've all should have all signed the forms for. And Sam has extra forms for if you've anything you haven't filled out. Please make sure you do it today and again give it back to us. Remember that you must not download anything from the server. That is something we promised the IRB, and we'll all get in a lot of trouble if you download anything. And you are responsible for data security and the privacy of the respondent. So don't, I don't think anyone would do this, but don't try to figure out who anyone is. That would be a violation of all sorts of rules. Okay, so I've already mentioned about faculty office hours. Let me just mention that a number of us who are here now are going to be here throughout the entire 2 weeks. So that's David, Patrick, and myself, Hui and Rosie, Andrea, who is going to be our expert on the biology, and James, who's going to be teaching population genetics, but as you heard last night, he's also he's an expert on everything.
Oh, I'm done.
Fix it. It doesn't help in this.
And Raymond, who is also pretty much an expert on everything. Basically, everyone's an expert on everything. And Raymond's going to be teaching LD score regression and related genetic correlation and related methods. Thank you. All right. So we're going to talk about the causal model, which is the general framework that's going to underlie, a unifying framework for thinking about everything we're going to talk about over the next 2 weeks. So we have some outcome of interest, which I'm going to call Y. In the context of genetics, we call it the phenotype, and it's subscripted by I for the individual. So that's, for example, individual I's level of educational attainment. And then our genetic data, I'm going to denote by X. So this is individual I's genotype at location J in the genome. So there's going to be a lot of J's. And each of these genotypes is a 0, 1, 2 variable. So that's one of the special things about genetic data that we'll be talking about why that's the case. But you can think of this as the number of minor alleles that you have, 0, 1, or 2, at a particular location. So the genetic data as a whole, this genome-wide data, is going to be your genotype at a number of these locations in the genome. So there's something like 3 billion locations overall in the human genome. Typically, our data is going to be more like 2.5 million locations that collectively capture much of the variation across people. So think of this data as a 2.5 million long vector of a 0, 1, 2 variable. And what we're generally going to be interested in is causal effects. So what we want to know is what would happen to the phenotype if you took an embryo who was just conceived and you changed their genome in some way, maybe at a single locus, maybe at many loci, but you make some modification, and then you see what is the effect of that modification on how the phenotype ultimately develops. Okay, so that's the hypothetical experiment that we have in mind. Now, if we just look at correlational data and try to don't actually do the experiment, but just look at how a genotype is correlated with the phenotype, that may end up being predictive, but not necessarily causal. So what are some reasons why that might be the case?
Parents' genes? Your genes correlate with parents' genes?
Yep. It may be, exactly. So your genes we correlate with your parents' genes, and your parents' genes affect the environment that you grow up in. Okay. That's another reason why, another example of a confound.
Maybe that one gene can be correlated with another gene that is causing the outcome.
Excellent. So one gene is correlated with another gene that's causing the outcome. Great. So let's have one more. Okay, so what's population stratification?
That certain alleles are more common in certain ethnic groups, for example, which might be related to your outcome for entirely social or genetic reasons.
Exactly. So your genotype is related to your ancestry. So different ethnic groups, say, have different genotype frequencies, and that's associated with all sorts of environmental factors that matter for the outcome. Okay, so there's lots of these reasons. So in order to have our ideal experiment where we're looking at something like this, we're getting the causal effect of the genotype, we either need to be studying a population where you really do the experiment, you actually randomly assign the genotype, and that exists with animal experiments, but typically not with humans. With humans, what we need to do is include control variables such that given those control variables, the genotype is as good as random. So there is a particularly good set of control variables you could have, which is the parent's genotypes. Because as we mentioned before, and we're going to talk about the biology, given parents' genotypes, the child's genotypes are actually randomly assigned. So that's a very good approximation of our experiment. Another thing we could do that's not as much information as knowing the parents' genotypes, but is just as good in terms of getting random assignment, is to have family fixed effects. So saying, these individuals are siblings from the same family. We don't know the parents' genotype, but knowing that they're siblings means that we can treat the differences across them as random. Another thing we could try might be conditioning on ethnic background. That's not as good as these. So why is that not as good? Yeah, within populations, there's going to be some of the kinds of confounds that were mentioned. For example, the confound that your genotypes associate with your parents' genotypes that affects their behavior, that affects the environment that you grew up in. OK. So in terms of the theory of what we're trying to get at and the interpretation that we're aiming for, we want to think of always having these conditioning variables so that we can think of our idealized experiment and talk about random effects. So that's always the model we're going to have in our minds. When we do the theoretical derivations, I'm going to sometimes omit the z's just to keep the notation simpler. But you should think of it as there. Now, when we come to data, this issue of whether we're controlling for a sufficiently good z is going to be a critical issue. And to the extent that we don't have things like family fixed effects or parents' genotypes, we should always be worried about confounds that are introduced by not having the random experiment. OK, so now we're in the world of deriving the theoretical framework. So we're going to imagine we have those kinds of controls. And we're going to define the genetic factors that matter for the outcome, that we're going to call the genetic factor for the phenotype, as some function g, which is the difference between the expected value of the outcome given that we know the genotype relative to what the expected value would be without knowing the genotype. So think of these expectations as across people in the population. This is the mean phenotype in the population, and this is the mean phenotype among people who have that genotype. So we can So once we define the genetic factor that way, note that we can always write the phenotype, d-mean. So we're going to make things simple, we're going to d-mean the phenotype. And we can always write the d-mean phenotype as the genetic factor plus noise. And because of the random assignment of the xi, the genotype vector is not going to be correlated with any other factors that matter for the outcome. And so the expected value of that error term, given the genotype, is equal to 0. OK? So that comes from the random assignment. Yeah?
To that point again, like the correlation with the noise given xi.
Sure. So the expected value of the noise given xi is saying, is there any predictability in the noise, if we knew your xi, could we say anything about what your noise is likely to be? The answer is no. We can't say anything about that. And the reason that this is true is because of the random assignment. Because x is randomly assigned, so there's so any other factors that that matter for so this is our best guess of the phenotype given our genotype. And so anything else, if it were predictive of the outcome, it would already be in this term. So actually, now that I'm thinking, this may not be you may not need random assignment even for that to be true. Although I'm thinking on my feet here. Let's see. Yeah, I think yeah, actually, I don't even think we need the random assignment. I think this is the predictable component. This G is the part that's predictable based on the X, and this is the part that's unpredictable based on the X. And so we can always write this. We can always decompose in this way.
I think you want the G to be related to the causal thing that we were talking about at the beginning.
Yeah, that's right. So the causal, the random assignment is crucial for this to be interpreted as a causal effect. Yeah. Thank you.
I have another question.
Yeah.
Here we're assuming there's one true genetic effect is the same for everybody, right? So there's no, G by E, for example, there's no differences in how your gene influence the phenotype based on, say,
No. So we're not assuming that. We're not assuming. There can be gene-environment interaction here. There can be gene-gene interaction. We'll see that in a moment. I'm going to go from this to show where those show up. But this is completely general. This is just saying, given some vector of genotype data and a phenotype, we can always define the genetic factor this way and always write the phenotype as the genetic factor plus noise.
Wouldn't be causal anymore.
It'll be causal as long as X is randomly assigned.
Do you think of G as a big function that has a lot of things written in there?
Yeah, that's right. The G is a general function that could involve interactions between the X's, and it could involve interactions with environmental factors. This error term is environmental factors that are not interacting with the genotype, or measurement error, or other kinds of things. Yeah.
But if we're looking at it within the family, then it is kind of, you don't need to account for interactions, right? Or do you?
Well, there could be interactions between.
But you can still claim that it's causal because it's fixed at the family level, no?
So I mean, this is so. So we're going to talk a lot more about. David's going to be talking about this actually a little later today. There are reasons why you might, different siblings within a family could be exposed to different. Different siblings could be exposed to different environments, and those environments could interact with genotypes. So that could end up mattering for the phenotype.
Yeah.
But nonetheless, this is interpretable as causal if you have if you're talking about siblings within a family. It's just going to be causal, but averaging over gene-environment interactions.
Yeah.
Yeah.
And of course, you're estimating the average treatment effect. The only correct causal inference there is differential fertility, or based on the genotype of the earlier child, or some sort of antenatal selection, where prenatal environment affects the survivability of a given genotype in utero, and then you get you don't have random assignment orthogonal to the prenatal environment. But other than those, you're just averaging over all the gene
these kind of selection effect, even within utero selection effects of the population that we ultimately have, this could be in principle written into that big function. Okay? In principle.
I feel like Dalton's point is kind of an empirical. So the issue is that if we have data and we just don't observe people with certain genotypes because they just don't survive, then we can't estimate it. Although I guess we could say, yeah, sure, we have this like G that exists in the population of every embryo that's ever conceived. Like, so if we're talking from a theoretical level, yeah. But then once we're trying to address it with the data that we have available, it becomes a bit tricky.
Okay, so this is a very general statement that we can always make. Now we don't typically work with this super general model with this super general G function, and the reason is because when we get to estimation, or one of the reasons is when we get to estimation, it's already going to be hard enough if we're dealing with a linear model to deal with all of the multiple hypothesis testing. But getting to a linear model is certainly helpful. We can also get so what I'm going to show next, and this is you'd work this out on the problem set. So this is review, is that you can get to the linear model also very generally. It's not imposing additional assumptions. Okay, so I'm going to just, from now on, I'm just going to use yi to refer to this demeaned y. So we're going to be focusing on what's called the additive component, which is really the linear component of G. So how do we get to that? Well, we write the best linear predictor of the phenotype. So this is not the best predictor. The best predictor would be the conditional expectation that defined the G function. Now we're taking the best linear predictor. So we're asking what is the linear function of xi beta, that's the best predictor in the sense of minimizing the sum of squares. So we're going to minimize the expected value of the squared deviation. That's going to give us our beta. And when we solve this problem, we get the population regression function. So we have some beta, that's the best linear predictor. And we know how to calculate that beta. It's the standard formula for ordinary least squares. It's the x prime x inverse x prime y factor. So note that we can take this G function, which we've defined this way, and always decompose it by adding and subtracting the best linear predictor. So we're not making any assumptions. We're just adding and subtracting the same constant. And then we take this part of it, which is well, let me start with the other part. We take this part, which is the best linear predictor of the phenotype relative to the population average level. And that's the additive component. So this is just subtracting off the mean of the population, just the linear piece. So that's the additive component. It's a function of xi. This is xi beta. And this other piece, everything else, all the rest of G, we're just going to call the non-additive component, n. So we can write this genetic factor as the sum of an additive component and a non-additive component. And we know that these have to be orthogonal to each other. Why? Because A is a best linear predictor, which means it's a population regression line. So anything which isn't on the population regression line is a residual. And by construction of a linear regression line, the residual is orthogonal to the regression. So n and A are orthogonal to each other. So what that means is that we can always, very generally, without making any assumptions, we can always write the phenotype as equal to the sum of an additive component, a non-additive component, and an error term, where the error term is mean 0, and the three components are mutually orthogonal to each other. Okay, so it's just a decomposition. Now, we could further decompose the non-additive component, and sometimes we do. The non-additive component could be decomposed into a dominance piece, which we denote by d, d of x. So that captures nonlinear effects of the genotype. So that would be differences of going from 0 to 1 or 1 to 2. And we could it also includes what are called epistatic components. Those are gene-gene interactions. So those are cases where the genotypes of two or more variants are interacting with each other. And that epistatic component, in turn, has many subcomponents that you could study. You can study pairwise gene-gene interactions or interactions of the additive component of two loci, or pairwise interactions between two dominance components, or pairwise interactions between the additive component at one locus and the dominance component of another, three triples of additive component interactions, and so on. Okay, so there's many different kinds of epistasis you could study. The dominance component, however, can be defined even for a single locus. So in the problem set, you did the case of a genome with just a single variant in it, a single variant model. And in that case, the non-additive component was the dominance component because there is no epistatic component. You can't have gene-gene interactions when you just have one locus. But more generally, the non-additive component has both of these. OK. So when we focus on the additive model, what we often do is group together the non-additive component and the error term and group them into a single error term, a composite error term. And since everything is mutually orthogonal, that's fine. Here, we're still going to have an error term that's orthogonal to the additive component, which we often write this way. And if we don't suppress the covariates, then it's written this way. We have the phenotype is this linear function of the genotypes plus our controls that we need to have a causal interpretation plus an error term. So in this additive model, we call the beta here the vector of the genetic effects. What's the interpretation of the beta? Well, we know it's causal, but it's an average effect. So it's the effect of a hypothetical experiment. So beta j is the average causal effect of a hypothetical experiment where you intervene at location j in the genome, and you change the genotype from 0 to 1 or from 1 to 2 at conception. It's an average in a number of ways. It's an average over the dominance, because it may be that going from 0 to 1 effect is not the same as going from the 1 to 2 effect. We're looking, when we do beta j, we're looking at an average in the population, a weighted average depending on the likelihood, the frequency in the population of people being 0, 1, or 2. It's also an average over whatever gene-gene interactions there might be. So it's given the genetic background in the population. And it's an average across individuals. It might be that the effect for me of going from 0 to 1 is different from the effect for you. So it's an average in many senses, but it's nonetheless has a causal interpretation. But because it's an average effect, and it could be that if you go to a different population with different levels of background environments that are being interacted with or background genomes that are being interacted with, it's actually a different number. So it's not a fixed parameter that is going to be the same necessarily from one sample to another sample. But within a population, it's a well-defined causal object. So it's like an average treatment effect in a randomized experiment. It's analogous to that. Okay, so if we don't write it as a vector, but we write it as in scalar notation, it's a sum. And we're going to discuss a number of extensions to this. So we're going to often study this additive model, and it's going to be particularly useful when we talk about genome-wide association studies, because this is the model that we're estimating. But then we're often going to want to move beyond the additive model as well, and talk about things like covariates, dominance effects, epistasis, gene-environment interactions, gene-environment correlation, and some other assumptions. But this is a useful framework for thinking about how everything relates to each other. So with that in mind, I'm going to move to the last topic and quickly talk about the outline of what the syllabus is going to look like over the next 2 weeks. So the first topic is the potential payoffs for social science. And we're going to particularly focus today on psychology and sociology because some of the applications at the end of the course are going to be economics. So you could ask, why put these lectures on day one when in fact they're going to, as you'll see, they're going to be drawing on some tools that you're going to be learning later in the course. But I think it's useful to have them on day one because their motivation. They're going to show you what we can hope to get out of all of this and why it's useful to learn about some of the tools later on. And also, to help you start thinking now about your own research ideas, I think it's useful to see concrete examples of applications of genetic data. Also, as part of the kind of big picture topic, Michelle's talks on the ethical issues are very important to keep in mind. Okay, so then the second topic, which we're also going to start today with David's talk on heritability, is behavior genetics. So behavior genetics is the term that's often used for twin studies or adoption studies or other kind of family-based studies, and it pre-existed genetic data. I mean, behavior genetic studies have been around for 100 years, and they're often an important motivation for studies that use genetic data. So they're still relevant now. The way to think about behavior genetics is that instead of studying the sum, the individual genetic effects, the betas, we focus our attention on the whole sum, the additive genetic factor as a whole. And we can infer things, even if we don't have genetic data. It turns out that using clever study designs like adoption studies or twin studies, we can infer things about the effects of this additive genetic factor, even if we haven't measured the genotypes themselves. The main goal of a lot of that work is to estimate what's called the heritability, which is the fraction of variance explained by the additive genetic factor as a whole. And it's useful to understand, partly because it's going to build on the same model and a lot of the issues that we just talked about very briefly in terms of interpretation and causation. David's going to go into much more detail about them in the context of behavior genetics. As we're going to talk about predicting phenotypes with genetic data, constructing polygenic scores, and the heritability is going to turn out to be an important parameter because it's an upper bound on the predictive power that we could ever hope to get with actual genetic data. And it's going to serve as a benchmark for other methods of estimating heritability that use the genetic data themselves. Okay, the next big topic, which is going to start tomorrow, and Andrea is going to give 4 courses on molecular genetics. That's going to teach the basic biology, which is important to understand when we think about the modeling assumptions that we make for the methods that we use. We'll understand when they're reasonable, when they're likely to fail. We'll talk about where genetic data comes from, how it's actually measured, so what it is that you're using, and the new kind of data that's going to be available in the coming years. We're going to talk about biological mechanisms by which genes matter, and how the environments interact with genes. So we're going to talk about gene expression, epigenetics, and that'll come up again as well later in the course. So then the fourth topic is how we know something about which genes relate to which outcomes, and that's called gene discovery. And that's important to understand because when you want to apply genetic data in the social sciences, you need to build on what we think we know about which genes relate to which outcomes, or what polygenic scores work. And it's important, if you're going to build on the work, to know which is reliable and which isn't. So how do you evaluate that? And the challenges that come up in the topic of gene discovery, power, multiple hypothesis testing, and population stratification, are the same big three challenges that arise in applications of genetic data. So we'll go into detail on those in this topic. The fifth topic, and this is really a collection of topics, but they all kind of could be categorized as exploiting the aggregate information contained in the genetic data overall. So one of the things we'll learn in the gene discovery part of the course is that individual loci have very small effects for the kinds of behavioral phenotypes that we're interested in the social sciences, but the joint effects of many of these loci taken together can be substantial. And the joint effects are again captured by this AI term. We can construct polygenic scores, which are predictors of this AI. So they're A-hats. There are estimates of the additive genetic factor. We can also exploit the aggregate information by calculating genetic correlations, which are correlations between the BJs for one phenotype and the BJs for a different phenotype. So it's asking how much overlap is there in the genetic factor between two phenotypes. And another way to exploit the aggregate information is methods based on partitioning. So if we want to learn something about say, biological pathways by which genes matter for an outcome, we can ask whether the beta J's tend to be larger for some types of loci that work through certain biological pathways relative to other types of loci, for example. There's various ways you can partition the data. The sixth topic, which again is really a collection of things, but it's all about environmental effects. So gene-environment correlation, gene-environment interactions, which is how environmental factors modify the betas or modify the effects of all the betas taken together, the A. Mendelian randomization, which is a way to use genetic data to estimate the causal effect of an environmental factor. And there'll be a couple of lectures on epigenetics, functional genomics, and life course development. So the big picture here is that much of what we want to use genetic data for in the social sciences is actually not about genetics at all. It's about learning about environmental factors. And there's a number of ways in which having genetic data can give us more leverage to do that. So we'll talk about a number of methods for that. And then the last topic is applications, where we're going to go into detail on several of the applications that have been done so far, explore some of your research ideas, and then talk about what methods are being developed or likely to be developed in the near future, and what are, we think, important directions in which social science genomics is going to move. So let me preview some of the key themes that are going to come up throughout the 2 weeks. One is that empirical results are only informative if the analyses are well powered. We'll talk about that at length, and why, in general, you probably shouldn't undertake a study in advance if you know it's not going to be well powered. we'll talk about the 3 central challenges I mentioned to gene discovery, but more broadly to applications of genetic data in social science, power, multiple hypothesis testing, and population stratification. And finally, we'll talk about how genetic effects often operate through environmental mechanisms, especially for the kinds of behavioral and social science phenotypes that we're interested in, but even more generally. And this is one of the reasons. Why genetic results are often misinterpreted and misunderstood, because people don't get this. They think they see genetics and they think biology, but that's not true. And it's also why genetic data. One of the reasons why genetic data can be so useful for the social sciences, because it can teach us about the environmental mechanisms through which the genes are operating. OK, so only 3 minutes over, which is pretty good given the technical difficulties. And we will now have a break and start up again at 11 o'clock.